Hello everyone, let's start with the elongations of the DNA replication. Now, DNA polymerase catalyzes a step-by-step addition of the DNTPs to the DNA chain, or we can say strands, and the two new strands are the opposite to synthesize. One is continuous synthesis, leading strand. We can call that as a leading strand, and the other is synthesized in the discontinuous fashion, that's called the lagging strand. So this is the difference between the leading and the lagging strand. The leading strand is continuously synthesized and the discontinuous one is the lagging strand. Now, looking at the lagging strand requires many RNA primers. These short fragments are called the Okazaki fragments. After 1000 to 2000 nucleotides of the leading strands have been replicated, after the first round completed, it continues the discontinuous strand synthesis. Now, the joining of the Okazaki fragments on the lagging strands to make a continuous strand, they require the DNA polymerase 1 and DNA ligase. As each Okazaki fragment's formation completes the RNA primers of the previous fragments and is removed by the 5 to 3 exonuclease activities, and then the fragments are joined to the continuous strand by DNA ligase. Looking at the leading strand synthesis, it is a 5 to 3 polymerization activity. After the addition of the several nucleotides in the daughter strands, primer is removed and the DNA polymerase ellipsin, due to its 5 to 3 exonuclease activity, it removes the primer and the gap is filled by the same DNA polymerase ellipsin. And the DNA polymerase delta is involved in the proofreading activity. The nick or the gap is filled by the DNA ligase. Looking at the lagging strand synthesis, the lagging strand synthesis of the Okazaki fragments to initiate the same way the leading strand synthesis. Now, it is a continuous fashion of the 150 to 200 nucleotides in one Okazaki fragment. Here, the RNA primase is synthesized by the DNA polymerase alpha due to its primase activity. Now, the DNA delta used as proofreading, the strand is synthesized by 5 to 3 polymerization activity and the gap is sealed by the DNA ligase. Now, here is the diagram of the elongation phase of the DNA replication. You can see the lagging strand, you can see the leading strand. All the enzymes that are involved, the topoisomerase, the primase, DNA ligase, DNA polymerases, the other proteins, MCM, CDC, RPA, and all other pCNA is also here is the helicase acting as the helicase.